Now, our next presentation is on Dr. Mohammad Abdus Sobul Talukdar. And Dr. Abdus Sobul Talukdar is the Assistant Professor of Agriculture, IUBAT. And you can
Hello, dear chair and co-chair and dear audience. Welcome to my presentation. This is Dr. Abdus Sobul Talukdar. Now I am working as the Assistant Professor, Faculty of Agriculture, IUBAT. This is my presentation title, Elucidation of Lysosomal Cathepsins in Bovine blood leukocytes during early pregnancy. Actually, this is the complex phenomenon after ovulation in a cow that it goes to fertilization with the sperm and then subsequently develop 2-cell, 4-cell, and 8-cell, and 16-cell, and then blastocyst. After hatching blastocyst, it creates a conceptus. Conceptus secretes on protein known as interferon tau, which subsequently gives signal to the maternal uterus, and subsequently implantation happen, and then placentation, and then fetal growth will be developed. Actually, what are the maternal recognition of pregnancy? It is simply the signal between the conceptus and then maternal system, actually. In ruminants, especially cow, buffalo, sheep, and goat, actually, they have 4 stomach and then complex reproductive system. And so after develop conceptus, it secretes interferon tau. That interferon tau gives signals to the uterus, and then uterus also gives signal to the conceptus. So this is the establishment of pregnancy. Several research has done during implantation, during conceptus growth, and then protein also as well as growth, and then mRNA protein also increased through interferon tau effects. This is my background slide. Actually, once upon a time, scientists think about the interferon tau secretes only into the uterine, not enters into the peripheral blood circulation. But now research demonstrates that interferon tau secretes into the uterus, and that through uterine vein it enters into the peripheral blood circulation. And from blood, after separation of the peripheral blood leukocytes, and also we analyze different genes that are stimulated during the early pregnancy time. That is IHG15, interferon stimulated gene 15, MX1, MX2, and OAS1. This is the highly potential gene expressed in the early pregnancy time. It's simply comparison between human and bovine. Actually, in case of human, what happened? Day 1 after fertilization, and then day 5 to 6 it hatch, and then after 6 days it attaches to the uterus. And after attachment to the uterus, then immunological recognition happen, and then finally pregnancy happen. But in case of ruminant, the conceptus is elongated, and then interferon tau protein secretion is increases at day 12 to 18, and then finally within uterus immunological recognition happen, and then establishment of pregnancy. Actually, I want to similar with Sheikh Shadi sir. If you search in the Google, there is any pregnancy detection kit for bovine. Actually, it's still not any detection kit in bovine, but it only in the human. But if you go any veterinarian or any other technologist, they can say easily, okay, we can test blood, no problem. We can test urine like as human. But human reproduction and bovine reproduction looks same, but it has several complex phenomena. In this research, I have published 3 papers. Actually, first paper I have published in the reproduction, it's the famous journal published from Cambridge, and then also another GRD, reproduction of development, and then Pakistan Journal of Biological Science. And last 2019, I have received from fellowship award from our Honorable Prime Minister. Actually, I am the fellow of the Bangabandhu Science and Technology Fellowship Trust for financial support. In this slide, just I want to describe about what are the early pregnancy diagnosis. Actually, improving the reproductive efficiency and pregnancy rate by decreasing the interval between EI service and increasing the EI service rate as well. Economic consideration of the dairy farmers, interference, and then policy makers, and change the management, feeding, and healthcare system of our farm to promote a healthy pregnancy and early pregnancy termination if so desired. Now, current status of the diagnosis of pregnancy, in our country and worldwide, rectal palpational ultrasonography is the famous technique, but it needs skill and experience in instrument. Only 30 to 40 days after insemination, risks of embryonic mortality. Later, actually, we have tried to develop immunobassy on progesterone and early pregnancy associated glycoprotein, but it has also limitations, sensitivity, specificity, and accuracy. But now, pregnancy biomarker molecules. In my research, actually, my final outcomes is the pregnancy biomarkers molecules, which is specifically up and down regulated in the blood, expressed over a considerable period of time, and present in the accessible body fluids and non-invasive. Just I would like to about what is cathepsin. Why I select cathepsin actually? So in the cell contain lysosome, and lysosome contain many proteases, lipase, and many things. So important proteases is the cathepsin, and now many research demonstrated that cathepsin actually is a vital factor to implantation of the bovine. So cathepsin is the lysosomal proteases that degrade of intercellular proteins in the lysosome. According to its structure and function, there are 3 proteases, but in my research, I used 16 proteases that are subsequently developed pregnancy and implantation have important functions. Now, 12 cathepsin are already identified in case of human. So, from my background study, now I decide my objective to explore the new biomarker molecules for pregnancy detection by investigating the activity of lysosomal cathepsins, expression of cathepsin genes, and detection of cathepsin protein in peripheral blood leukocytes. Now I conduct 3 experiments, effect of interferon tau, and measure the activity in the peripheral blood monocytes and peripheral mononuclear cells. That means monocytes and granulocyte cells, and expression of genes, and then immunohistochemistry for detection of the cathepsin B protein. Now, this is my simply material methods. I have in the dairy cows, in vivo and in vitro experiment, I have collected blood and processing day 0, 7, 14, and 18 after artificial insemination and separation of monocytes and granulocytes, and immunohistochemistry, sampling for RNA extraction, subsequently gene analysis by RT-qPCR, and then detection of cathepsin activity, that is magic fluorescent activity and data analysis. And also in vitro collection, I have collected blood from non-AI, that is control group of the animal, and also separation of the monocyte and granulocyte. And then I have administered recombinant bovine interferon tau, which is secreted from the conceptus. And then also I incubated several hours, and then I have detection activity, RT-qPCR, and then immunohistochemistry, and finally analyzed the data. This is simply the cathepsin BKL activity in pregnant and non-pregnant cow. Actually, you see cathepsin B monocytes, it almost non-significantly different, but granulocyte, J18, it increases significantly, and also both pregnant and non-pregnant, which show significant difference. Same cathepsin K also, both monocytes and granulocytes also, day 7, 14, and 18, is significantly upregulated, and also both non-pregnant and pregnant is also significant. And also, cathepsin L also significantly upregulated in different days. So this is the simply in vitro results. Actually, this is the control group, and then this is the interferon after administration of interferon tau, and I incubate different hours. And so it looks more fluorescent intensity than control group. It's quite difficult here, but when I put the image in the fluorescent microscope and image analysis software, and we can differentiate very clearly. Simply, this is the in vitro induction of the interferon tau. So after in vitro induction in different hours, it also increases, suppose 12 hours, and here 6 and 12 hours significant. And cathepsin K also 6 hours and 12 hours it also significantly. And then here also significantly increases after in vitro induction of the interferon tau. This is simply RT-PCR. First, before I do RT-PCR, I have tested the all genes. These are present or not in the peripheral blood lymphocytes. So I have found all genes are significantly expressed in the all pregnant and non-pregnant cows. This is very vital information. IHC15 actually now in the world, actually most scientists think about this gene. This is the very vital genes for the developed a biomarker kit. Actually, this is the IHC15, interferon stimulated gene 15, which also regulated day 18, both monocytes and granulocytes. And also cathepsin B genes, this is my genes also that is also regulated in the day 14 and day 18, both pregnant and compared to non-pregnant cows. Also in vitro experiment after induction of the interferon tau, so these also increases significantly after in vitro induction of the interferon tau. And immunohistochemistry for detection of the cathepsin B protein, as we know, cathepsin B is a very low molecular weight protein. It's quite difficult in blood, actually in a large animal, it's a very luminous plant. So it's quite very difficult. So in my research, I did over 6 months to get such kind of the image. Every day I have performed immunohistochemistry, Western blotting and such and such. So it also I have found in pregnant compared to non-pregnant, pregnant show more fluorescent intensity than non-pregnant, and statistically it is significantly increased both monocytes and granulocytes. And after in vitro induction of the interferon tau, the interferon tau group show more fluorescent intensity and is statistically significant. So now I'd like to conclude my research. Actually, type 1 interferon tau is a possible inducer of the cathepsin activation in monocytes and granulocytes. Cathepsin B protein expression and concentration in monocytes and granulocytes is affected by pregnancy. And cathepsin genes in both monocytes and granulocytes would be useful target gene for reliable trajectory of pregnancy before the maternal recognition of pregnancy cows. So this is my outcomes in my research. Actually, I have planned in my research group in Japan, Hokkaido University, my sensei and me try to establish a detection kit. Actually, এখন পর্যন্ত এখনো এটি রুমিনেন্ট অ্যানিমেলের কোনো ডিটেকশন কিট আবিষ্কৃত হয়নি। মানুষের যেরকম আমি 6 দিনের মধ্যে এটি কনফার্ম করতে পারছি সে প্রেগন্যান্ট কিনা এটি ইউরিন টেস্ট করে, কিন্তু এখন পর্যন্ত বোভাইনে এটি এখনো সম্ভব হয়নি। বিধায় আমাদের এটি টার্গেট রয়েছে অ্যাজ আর্লি অ্যাজ মাস পসিবল টাইম আমরা এটা ফার্ম টু ফার্মে আমরা করতে পারবো। আশা করছি যদি এরকম একটা কিট করোনা ভাইরাস যেভাবে ডিটেক্ট করা হচ্ছে এরকম কিট যদি আমরা করতে পারি, এটি আমাদের রিসার্চের সবচেয়ে বেশি ইম্পর্টেন্ট দিক হবে। I have also published many papers. This is part of this papers. And now I have acknowledged my Bovine Science and Technology Fellowship Trust, Ministry of Science and Technology, and Supervisor Masashi Takahashi of Hokkaido University. Thank you very much. Thank you for your kind attention.
Thank you for this. Thank you, Dr. Abdus Sobul Talukdar. So, any question of the audience?
Suggestion, comments. This is thank you very much. This is Ms. Bahut Jaman from Secretary of Agriculture and Division. Very much interested. Actually, bovine is the large dominant of the basic population. You know, buffalo, maxi, sheep, goat, cow, etc. So this is excellent presentation. Thank you, Dr. Saurabh Talukdar. Highly appreciate your work, and further, just I would like to request you to work on the FMD control system research in collaboration with the veterinarian. So now the long-skin disease is very emerging in talent. It's very to get safety, food safety from the slaughter. And thank you very much.
Okay, thank you. But actually, one thing that I want to know. Normally in human cases, we use the urine and teats. So is it possible for cows to use the similar type of?
Yes, thank you, sir. Thank you. আমি একটা স্লাইডে দেখিয়েছিলাম যে হিউম্যান এন্ড বুভাইনের যে প্রেগন্যান্সি এস্টাবলিশমেন্ট যে ডিফারেন্সটা human in case of human HCG human chorionic trophin hormone quickly secretes into the blood and then quickly passes to urine. That's why 5 or 6 days after pregnancy, women can check the STD test and she can get positive or negative result. That time actually animal scientists they are more emphasized maybe bovine এই ধরনের কোনো molecules আছে যেটি আমাদের সার্চ করতে হবে তখন তারা ইউরিন এবং পসিবল সোর্স মিল্ক ইউএসএ তারা এটি নিয়ে কাজ করলো কিন্তু তারা আমি যে জিনিসগুলো ব্লাডে সিগনিফিকেন্টলি পাইলাম এগুলো তারা আসলে সিগনিফিকেন্টলি পায়নি একটা দ্যাটস ওয়াই তারা এমফেসিস দিল যে ব্লাড কারণ আপনি জানেন স্যার যে এখন ব্লাড আসলে সবগুলো ডিজিজের ক্ষেত্রে আপনার টিউমার হয়েছে মাথায় বা ব্লাড টেস্ট হচ্ছে আপনার কিডনি হয়ে পাথর আপনার ব্লাড টেস্ট হচ্ছে সুতরাং সবগুলো জিনের কিন্তু সিচুয়েশনটা এখন ব্লাডে আপনার নরমাল কন্ডিশন এন্ড এফেক্টেড কন্ডিশন এগুলো কিন্তু সিগনিফিকেন্টলি এবং এগুলো কিন্তু ক্লিয়ারলি এখন শো করছে যার কারণে ব্লাডটাকে আমরা আমাদের র মেটেরিয়াল হিসেবে আসলে
Thank you. You got this answer.
থ্যাংক ইউ স্যার থ্যাংক ইউ